So first of all, I just wanted to start off with a plug and introduction in general for the EPAASM student chapter, especially for any students who might be online and interested in who we are and what we do. So our faculty advisor is Dr. Sunny Shin, who unfortunately couldn't be here in person today. And like I said, Marissa and I are the co-presidents. We also have our EPAASM treasurer, Simon Knight, who is always incredibly instrumental in helping us out with various financial aspects that we don't really understand or know what's going on very well. Dr. Michelle Kutzler is, of course, our EPAASM president. And then we have our student officers. So Marissa and I from the University of Pennsylvania. This year, we had a really great student board. So we had our vice president, Caitlin Grando, our treasurer, Zach Walter, who's actually one of our speakers today, and then our secretary, Benjamin Haslin-Gorley. And so in addition to having a variety of different talks, pictured here is one of our previous May monthly meetings, I think from back before pandemic times, the last time we were really lucky to be able to get together in person. The student chapter is really lucky to have the opportunity to organize the Philadelphia Infection and Immunity Forum in December, which is a great opportunity to bring together microbiologists, immunologists, in sort of the greater Pennsylvania area for a really great day of talks. So that's something to also look forward to. And if you're interested in getting involved, please contact one of the student officers. Additionally, we occasionally have fun science-oriented excursions. So this was a photo from a science after hours trip at the Franklin Institute, which is always a really, really great time. And most importantly, for students to join the student chapter, it is completely free. And so there's really no downside and only really, really great benefits to becoming a part of it.
Thanks, Jenna. Yeah, so as Jenna was saying, we love the EPAASM chapter. If you want to find out more information, there is a website that has all of the information about our chapter and then also upcoming events. So we will have a meeting in June. That's exciting. And then it also outlines the different member benefits. We'd really like to encourage, especially students, to join the chapter. Like Jenna said, it's free. And there's a few different benefits. The biggest one we believe is networking with other microbiologists in the Philly area and really joining this community. And then you'll also receive monthly announcements about the different events that are sponsored by the branch, and then even have a discounted registration and other fees for special programs. So a lot of benefits. And if you are interested in becoming a member, please reach out to any board member for more information.
Okay, great. And so this is sort of the close of the semester. And so we're going to have a turnover in the board. And so Marissa and I are really, really grateful for our time working with the EPAASM student chapter board. I know Marissa has been a part of this board for a really, really long time, pretty much her entire time as a grad student in Penn. And I was really lucky to come on this past year. So we're really grateful to have had the opportunity to serve on this board. We also want to thank Ben, who's not here in person today, but has also served on the board for this semester and will be leaving. And we wanted to take this opportunity to introduce the makeup of the new board for the next semester. So the incoming co-presidents will be Caitlin Grando and Shingo Besso from Temple University. Zach from Jefferson will be stepping up as vice president, and we're really excited to welcome Adam from Drexel University onto the board.
Yeah, then we have a great lineup of speakers today, both the trainee talks as well as a great keynote address. So the first trainee talk is going to be from our very own Zach Walter, who will be talking about some of his thesis work on flaviviruses at Jefferson University. And then the next trainee talk will be Julie Joseph from Drexel, who will also be talking about a viral protein in the context of neuroinflammation. And just to mention that these were some of our students who actually won poster awards at TIFF this past December. So we're really excited to hear more about your research. And then we are going to have a nice little reception out in the lobby where there will be food and refreshments. So please feel free to stay for that and mingle. And then afterwards, we're really excited to have our keynote seminar Dr. Megan Povolonis from Villanova University is going to chat about her research program at Villanova, focused on a specific parasite, as well as her academic journey, which is really exciting. So we thank Megan for coming here from Villanova, and we're really excited for awesome training talks.
Great, so I'll hand it over to Caitlin, who will introduce Zach.
All right, welcome everybody again. So our first talk today, as Marissa said, is going to be our own Zach Walter from our student board. Zach is a PhD candidate at Thomas Jefferson University, and today he's going to give his talk called Post-Translational Modifications of Flaviviral Proteins, Modulate Viral Entry and RNA Replication. So he's just getting that set up now, but thank you, Zach.
All right, great. Can you guys hear me okay? Going through, all right. All right, thank you, Caitlin, for that introduction. It's nice to see everybody that turned out today. My name's Zach, and I'm gonna talk to you today about some of my thesis work in Holly Ramage's lab, working on, maybe we should let the people on Zoom know what I'm working on. All right, great. Working on post-translational modifications of viral proteins and their impacts on viral replication. So Holly's lab's interested in studying virus-host interactions during infections with members of the viral family Flaviviridae. In particular, we work on members of the genus Flavivirus, and my project primarily focuses on West Nile virus. Yeah. Oh, is it doing the presenter view? Okay, thanks, Vincent. So yeah, we're interested in studying virus-host interactions, in particular, mapping protein-protein interactions using proteomics approaches, as well as monitoring changes to the host proteome. So a little bit of background about flaviviruses. These are enveloped viruses. They have a positive sense single-stranded RNA genome that encodes 10 proteins. There's 3 structural proteins that make up the viral particle. Those are highlighted here in orange, and 7 non-structural proteins that are only expressed inside the infected cell. And these play important roles in reorganizing the host cell to facilitate viral replication. And this group includes a couple really important members that are vectored by mosquitoes, including dengue, Zika, and West Nile, which is what my project mostly focuses on. So West Nile is globally distributed. It was originally isolated in Uganda in 1937, but since then it's been detected on every continent except for Antarctica, and it's spread by mosquitoes. The enzootic cycle of West Nile virus in nature, it exists cycling between the reservoir host, which is birds, and then when mosquitoes bite infected birds, the mosquitoes themselves get infected. And if an infected mosquito bites a dead-end host, like a human, it can cause febrile illness that in a subset of patients progresses to a more severe neuroinvasive disease. In the United States, this virus was introduced for the first time in New York in 1999. It's since spread across the entire continental United States, and it causes annual outbreaks with thousands of cases and hundreds of deaths. So as you can imagine from that life cycle that I just walked you through, there's multiple tissues that this virus infects, and it has to interact with host factors in all of these different cell types. And then similarly, there are multiple steps that the virus has to undergo in order to successfully complete a round of replication. So it has to attach to and enter a cell. It needs to fuse with host membranes to release its genome, initiate replication, replicate its genome, and then make new particles and secrete them from cell. And something that our lab's interested in studying is the impact of host factors on this viral replication process. So at every step of this life cycle, the virus interacts with both proviral and antiviral host proteins that can either facilitate infection or restrict it. And from the virus's point of view, you want to basically inhibit the activity of antiviral host factors while facilitating interactions and activities of proviral host factors. And one of the ways that viruses can do this is by mediating the addition of post-translational modifications onto host proteins, which is something that our lab was interested in studying. So in pursuit of this, we established a collaboration with Jeff Johnson from Mount Sinai. We did in vitro infections with West Nile virus, and then used post-translational modification enrichment protocol to enrich for either phosphorylated peptides or ubiquitinated peptides from infected cells, and then used mass spec to identify what host proteins these modified peptides were from. But as kind of a coincidence, I guess, viral proteins also, of course, get expressed in the infected cell. So this also gives us a lot of data about modifications on the viral proteins themselves, which is something that I was interested in studying. So when we did this mass spec, we picked up both phosphorylation and ubiquitination sites on viral proteins. So when I joined the lab as a rotation student, this is something that I was really interested in studying. So first, I'll walk you through some data that we have about a site that we found that is ubiquitinated during infection that we think might play an important role mediating the early stages of viral infection. So here we have just a summation of all of the different sites broken down by viral protein of how many ubiquitination sites we found on each protein. So you can see on the structural proteins here, in particular the E, which is the dominant glycoprotein of West Nile, we found a large number of ubiquitinations on this protein. And the other thing we wanted to do is take the protein sequence of West Nile virus and then compare it to different species of flaviviruses. So basically doing comparisons to find residues that are both modified and highly conserved among different viruses, with the idea being that if this residue is conserved and it's modified, that modification might play an important role in the viral life cycle. So in an attempt to specifically study the post-translational modifications at this site, we use a site-directed mutagenesis approach to prevent specifically the modification. So ubiquitination normally happens on lysine residues. There's a covalent bond between this epsilon amino group on the R chain. But if we substitute lysine with arginine, it maintains the overall size, the charge at physiological pH, but it can't be ubiquitinated. Any substitution also can have off-target effects, of course, but this is a good way, the best way we have currently to specifically prevent ubiquitination at only one site. So in some data I'm not showing here, we found, I took all of those different sites we found to be modified. I made single site K to R substitutions, and I found that all of those viral proteins they express, all of them fold properly. They're all assembled into virus-like particles. They're all secreted out of the cell. So it doesn't seem like ubiquitination is important for any one of those steps in infection, but we wanted to know whether there was an impact on the actual infectivity of the virus. So to do that, we use this approach that's well established for flaviviruses called reporter virus particles. I find it useful to think of this like a pseudotype assay. So basically they're single cycle infectious virions. They can get into a cell via the normal early steps of infection, but they package a reporter RNA that's deficient in spread. It has a deletion in the structural protein, so it can get in and amplify the RNA and make a reporter, but it's not able to spread from cell to cell. So this allows us to, in a single round, assess whether there's a defect and sort of getting in and establishing infection. So the way they do this is by taking the viral genome, you break it up into two plasmids. One of them is just the structural proteins. The other one encodes the non-structural proteins fused to a reporter. If you co-transfect this into a packaging cell line, you get those single cycle viruses out, and then you can use them to infect target cells and measure the reporter. And since this is a plasmid-based system, we can use site-directed mutagenesis to make all of those K to R substitutions at all the sites we found to be modified and determine if there's an impact on infectivity. So this is the summary of a lot of work. This is what I did as a rotation student in Holly's lab. So you can see wild type are easily able to get into cells, and they make this reporter at really high levels, especially compared to our negative controls. And one mutant that I made, this envelope K110R substitution, has a really strong, about 98% decrease in infectivity. It still infects cells. It's higher than the level of our negative control, but it displays a really strong defect in infectivity. So the first place that you would think this might have an impact is at the level of attachment and entry into the cell, since this glycoprotein mediates entry. So I developed this internalization assay. We synchronize infection at a low temperature to keep the viruses outside of the cell, and then we shift it to 37. The viruses start to be endocytosed, and then we digest away the virions that remain on the outside with proteinase K and use qPCR to measure the internalized viruses. And you can see there's a statistically significant decrease in internalization when we make this substitution, but it's only about 40%, and that's not enough to account for the difference that we see in this reporter. So I did an experiment where I basically compensated for this by adding more virus. And if you give them an equivalent amount of virus that gets internalized, you still see this defect in the reporter that gets made. So that suggests to us that while attachment might be a part of the story, it's not the whole thing. It's probably also functioning at a post-attachment step. So the next step is fusion and uncoding. This is where the project is right now. I'm working on developing some in vitro fusion assays, as well as looking at syncytia formation in infected cells and recovering mutant viruses and characterizing their growth characteristics and pathogenicity. But this is something that all of this work is in progress now. So to shift gears and move from away from ubiquitin and towards phosphorylation, our mass spec also identified multiple sites of phosphorylation, including on the NS3 and NS5 proteins seem to be the most modified. And it's important to point out that these are the only two enzymes that are encoded by the virus. Everything else doesn't have enzymatic activity. So first, the NS3 protein, this is the viral RNA helicase and the viral protease. And it's really important in RNA replication. So we can measure RNA replication in the cell by using something called a subgenomic replicon. This is also deficient in spread. So instead of having the structural proteins at the 5' end, you replace those structural proteins with just a reporter that's fused to all the RNA replication machinery. So if you transfect this into cells, you get a lot of RNA replication, but no cell-to-cell spread. So it just lets us measure RNA replication. And similarly, we can make substitutions at our modified sites that we found to be phosphorylated this time and see if there's an impact on RNA in the cell. So we use mutagenesis again. So when serine gets phosphorylated, this hydroxyl group here is covalently attached to a phosphate, which also adds negative charges. We can either prevent that negative charge, the addition of those negative charges, by changing the serine to an alanine. No hydroxyl group, can't get phosphorylated, or by changing it to an aspartate, which at physiological pH is constitutively negatively charged. So this is what we call a phosphomimetic. So this is exactly what we did for the sites we found to be phosphorylated in the NS3 protein. You can see our wild type replicon replicates to high levels compared to our negative controls, which are a polymerase, a dead mutant, and a protease catalytic mutant. And when we make either phosphonull or phosphomimetic substitutions at, say, this site that's in the protease domain, we didn't see an impact on viral replication. But for one of our sites, if you prevent phosphorylation by mutating the serine 1972 to alanine, you get a really, really steep decrease in the amount of RNA replication in cells. So this substitution is in the RNA helicase domain. Since these are single-stranded RNA viruses, when they replicate their genomes, they form a double-stranded RNA intermediate, and it's the job of the viral helicase to peel those double-stranded RNA intermediates apart so that the replicated positive strand genomes can get packaged and go on to make new viruses. And this is absolutely required for efficient replication in the production of new viruses in the infected cell. So since this is such an important step in replication, the NS3 protein is really highly studied, and we reached out to someone who's an expert in modeling NS3, Dr. Martin McCullough at Oklahoma State University. So he took the sequence of the West Nile NS3, mapped it onto a previously solved structure for the dengue virus NS3, and then highlighted our amino acids of interest. So serine 466, that's its location inside NS3. 1972 is its designation in the entire polyprotein. So this is the important region for this substitution. And if we zoom in a little bit, you can see that this serine 466 residue is very close to the ATP binding pocket. Now that RNA helicase activity of NS3 is dependent on ATP. So we think that this residue here might be involved in a network of amino acid interactions that are stabilizing the ATP binding pocket and possibly cycling between ADP and ATP binding for helicase activity. So we're currently waiting on some modeling from this collaborator to see both what the phosphonolyl and phosphomimetic substitutions would look like at this residue, as well as what an actual phosphoserine would look like, and see if we can get any structural insights on ATP binding or possible RNA helicase activity. And we're also working on getting an in vitro RNA assay off the ground in our lab. Moving on to the last little bit of my talk, which is about a couple sites we found on the NS5 polymerase protein. So there are two major enzymatic regions of NS5. There's an N-terminal methyltransferase and then a C-terminal RNA-dependent RNA polymerase. So we use the same replicon-based approach that we did before at all these different sites to measure RNA replication. And you can see we found two sites, actually both of which are outside of the catalytic regions of the protein, which I thought was pretty interesting. And in this case, the opposite of what we saw from NS3, it actually appears that phosphorylation at these sites is inhibitory to RNA replication. Now, this is really exciting to us because this has been pretty well described for a related virus in a different genus in the same family. So hepatitis C virus, it's known that the NS5A viral polymerase gets hyperphosphorylated at a late step in infection, and that shuts down RNA replication because the virus wants the cell to transition from using its resources from making more viral RNA to packaging and secreting new viral particles. So this hasn't really been previously described for flaviviruses, but we think this might be early evidence that there could be sort of an analogous role for phosphorylation. But we are also working on getting an in vitro RNA replication assay off the ground to actually show that. So that's all I've got. So hopefully from this, I've slightly convinced you. Number one, we can use mass spec to identify PTMs on viral proteins. Number two, these proteins are extensively modified, including in functional regions, and also that modifications on conserved residues are probably playing important roles in the viral life cycle. So with that, I'd like to thank all the members of my lab, especially Holly, who's been exceedingly patient and understanding as we get all of these assays up and running off the ground because we're a new lab, as well as all of the current members and previous members, including Andrew, who contributed to this work, as well as the members of my thesis committee, our collaborators, Sarah Cherry, and particularly Jeff Johnson and Martin McCullough, other members of the flavivirus community for reagents and funding for both our lab and for me particularly. So thank you, and I'll take any questions.
I was curious about the envelope information. Is there, where it was in terms of the modeling the envelope protein?
Where on the protein? Yeah.
Whether you can explain.
Yeah, for sure. So that K110 residue, it's conserved in every flavivirus that I have looked at, that I've done the alignments for, and it's right smack in the middle of the fusion loop. So that's the region of the glycoprotein that extends out and it inserts into the host endosomal membrane, and then the viral and host membranes get pulled close together, and that's what allows for the release of the genome. So whether when we make the substitution and we get rid of the ubiquitination, I don't know whether the UB at that residue would enhance or would repress those membrane interactions. In my mind, ubiquitin is a very large modification. It's like 8.5 kilodaltons, and I would think that that would get in the way of the membrane interactions. But yeah, so for that particular site, we're expecting it to function at the level of fusion because of its location in that region of the protein.
Any other questions?
Okay, thank you.
Let's see.
Okay, you're not too. It's my pleasure to introduce our second training speaker, Julie Joseph from Drexel. And she's going to give a talk titled Retroviral VZIP Protein Contributes to the Release of Soluble and Exosomal Immune Checkpoint Molecules in the Context of Neural Inflammation.
Well, good afternoon, everyone, and I just want to take a moment to just thank the EPAASM members and board for giving me a chance to. Sure. Can you guys hear me better now? Okay. Yeah, just want to give my thanks to the board members as well as the meeting committees for the opportunity to speak today. So today I'm talking about a part of my thesis work, which is investigating the role the retroviral VZIP protein, also known as HBZ, and how it contributes to the release of soluble and exosomal immune checkpoint molecules in the context of a neuroinflammatory disease. Briefly, just an outline of my talk. I'm just going to be introducing HTLV1 and its associated disease, HAM-TSB, as well as what soluble and exosomal ICPs are, some characterization studies and studies on functional exosomes, as well as the effects of these immune checkpoint receptors that we're seeing in response to interferon gamma treatment, as well as antiretroviral drug treatments, and how this kind of led us to linking the viral protein HBZ to a possible mechanism associated with the release of these soluble and exosomal immune checkpoints, and some future directions we're considering based on what we have so far. So HTLV1 is actually an ancient or historic retrovirus. It stands for human T-cell leukemia virus type 1. It's an RNA retrovirus discovered in a patient in 1980 with cutaneous lymphoma, and it's considered a neglected tropical disease. And there's really an unmet need for both vaccines and treatment strategies against these neglected viruses and its associated diseases. It's often transmitted through bodily fluids and risk factors for intravenous, and is a risk factor for intravenous drug abusers. And historically, it was seen to be transmitted through tissue and blood transplants, but that's no longer the case anymore, as it's regularly screened before transplants. Most of the pathologies associated with symptomatic carriers end up being fatal, and there again is really no defined treatment, a cure, or vaccine against this virus. Now, it's estimated to infect anywhere from 10 to 15 million individuals globally. However, this is a gross underestimation because there's really no serological screening that's regularly done across the globe to really get accurate numbers. And in 2017, it came up that approximately 40% of Aboriginals in Australia were actually positive for HTLV1 virus. And this kind of really reinvigorated a lot of research and efforts towards eradicating the virus or finding cures and strategies for its associated pathologies. Now, just briefly talking about the overall pathogenesis of the virus, there's two routes of infection, either mucosal or blood. And once the infection enters the bloodstream, it eventually migrates towards your lymph nodes, where then it becomes an acute infection. And then from there, it progresses towards the bone marrow, where it can then infect progenitor cells and kind of persist within the body. Eventually, as the infection becomes chronic, 2 to 5% of individuals can develop adult T-cell leukemia or lymphoma, or 2 to 3% of individuals can develop HTLV-associated myelopathy or HAM-TSB. Now, there is 80 different clinical syndromes that are associated with HTLV1 infection, and what's kind of jarring to me is that HTLV1 infection, even though the global burden of it is actually very low, the lifetime risk of malignancy associated with infection is rather high compared to other viral infections. So my I would say, disease of interest associated with HTLV-1 infection is actually HTLV-1 associated myelopathy, also known as tropical spastic paraphoresis. Again, it can develop in 2 to 3% of infected individuals, and it's often characterized by proviral load and host susceptibility genes, and diagnosis is often delayed due to slow development. And it's often misdiagnosed as MS because a lot of the pathology that is seen mimics a lot of MS phenotypes. There's no clear understanding on how ham-TSB progresses, but it is considered to be an immune-mediated response. And again, there's no real therapy that's conclusively shown to really alter any long-term disability associated with ham-TSB, and clinical improvements, even though have been reported, is really based on a case-to-case basis using either corticosteroid treatments, interferon therapies, or plasmapheresis. Sorry, and the figures that you see right here is just depicting typical phenotypes that's associated once ham-TSB progresses, where you're seeing abnormalities in the white matter within the brain, as well as lesions in the spinal cord, and then perivascular infiltration within spinal cord tissue. Now, a unique feature of ham-TSB is that more than 30% of CTLs are actually specific to this viral transactivator protein tax, and that's really what most of the immune-mediated responses are targeted against. And although that response controls disease progression early on, it eventually, the CTL response eventually fails, and it contributes to inflammation and eventually bystander cell damage that ends up causing a heightened level of inflammation within the CNS. Now, both diseases, both ATL and ham-TSB, have robust responses against tax, but epitopes against other viral proteins like the envelope protein, as well as REX and HBZ, have been shown to be immunogenic. Now, before I jump into what my data shows, I just wanted to give a brief review of what immune checkpoints are. We know that immune checkpoint receptors are expressed in naive T cells like BTLA and TIM3, and then as T cells engage with an antigen, they become activated, and these immune checkpoint receptors are upregulated. And with the clearance of an antigen, you see a downregulation of these immune checkpoint molecules, but with persistent antigen, you see a continuous expression many of these immune checkpoint molecules that then eventually lead to an exhausted T cell. And a cohort study on the co-expression profile of both antigen-specific CD8 and CD4 cells saw that patients with ham-TSB actually have a higher frequency of quadruple positive immune checkpoint receptors that are present on the surface of their T cells compared to just asymptomatic carriers or uninfected individuals. And you see that significantly more so with multiple immune checkpoints being expressed in both CD4 T cells and CD8 T cells. Okay. So this kind of led me to think about my thesis project, which is investigating the role of how these soluble immune checkpoints are actually impacting some of the T cell responses associated that or contributing towards some of the T cell dysfunction that we see in ham-TSB patients. So soluble immune checkpoints are actually structurally and functionally similar to their membranous isoforms. They just can act on a systemic level instead of just locally with cell-to-cell surface engagement. And these soluble immune checkpoints are also present on the surface of exosomes, which are small extracellular vesicles that range anywhere from 50 to 300 nanometers in size. And exosome biogenesis typically starts within the early endosome, where then it can mature and incorporate different cargo from the Golgi and the cytoplasm to then become a late endosome and eventually mature toward a multivesicular body that is then secreted out of a cell. And these exosomes can carry a plethora of different molecules and proteins, including adhesion molecules, PD-L1, for instance, as an immune checkpoint receptors, as well as other metabolites and proteins. Now, there's actually been a huge presence of these exosome-related stories, so to speak, that have shown that these exosomal immune checkpoints actually have an immunosuppressive phenotype. And in HTLV1 specifically, we've seen that these exosomes can actually carry viral proteins like TACs and can actually contribute to overall viral spread and diminishing responses of healthy T cells and DCs. So to start off, I began to screen ham-TSB patients against asymptomatic carriers and serologically negative controls just to see what soluble immune checkpoint receptors are actually being present in their sera. And we saw both stimulatory and inhibitory receptors like CD27 to CD28 to be elevated in ham-TSB patients, but more so a lot of inhibitory immune checkpoints like LAG3 and PDL2. Now, unfortunately, due to the limitations of samples or specimens available in North America, I then switched over to a HTLV1 infected system of using 4 different T cell lines, Jurkat cells, ATLED cells, MT4 cells, and OSP2 cells. Jurkat as my uninfected control, ATLED and MT4 as more leukemic cell types with unique features, ATLED being only able to express the HBZ protein, whereas MT4 can both produce productive virus and pushes out a lot of TAX protein which has been shown to really drive a lot of the pathology of HTLV1 early on. And OSP2 cells is really the only established cell line associated with ham-TSB. So we saw that all these cells are CD4 positive, which is that tropic cell line associated with the retrovirus infection, and that MT4s and OSP2 do comparable amounts of TAX. So from there, I decided to just screen what soluble analytes are actually present in my cell systems. And similar to what we saw with the human sera data, the OSP2 cell line, which is that ham-TSB-like cell line in particular, we found elevated levels of BTLA, LAG3, PDL2, and IDO, which are all, again, inhibitory checkpoint receptors. So from there, I then decided to then characterize the exosomes that I'm then isolating from the cell line. So this is just my overall protocol associated with exosome exosome as well as characterization with Western blotting, just staining for typical canonical tetraspanins associated with exosomes, ALEX, CD63, and CD69. Calnexin is a negative marker associated with exosomes. You really only see it within the ER of a cell. So if it's present in your exosomes, you're not getting clean isolations. I also did an NTA analysis where we're really tracking particles that we're isolating to see whether or not they fall within the range of exosomes. So we see that with all 4 cell lines, we are able to isolate vesicles or particles that fall within the range of 50 to 300 nanometers. And what we thought was interesting here was that the uninfected cell line, you had a heterogeneous population of different sized vesicles where you're not really seeing that with the infection associated cell lines, where all of the vesicles that are being secreted fall within a distinct range. I then looked to screen to see what exosomal, immune checkpoint analytes are actually present in my exosomes. And again, we saw a similar pattern to what we saw with human sera, where there was elevated levels of BTLA, LAG3, PDL2, and TIM3. We also did some co-localization studies associating these immune checkpoint presence in escort proteins, which are TSG101 in particular, which is known to play an active role in recruiting cargo for exosome secretions. And we saw that particularly in ATLED and OSP2, that there was a distinct co-localization between PD1 and BTLA along with TSG101, that escort associated protein, showing that this is not just a passive process. These immune checkpoints aren't just being secreted out passively, but it's actively engaging within that endosomal sorting complex and being pushed out. And you're not really seeing that with that uninfected control cell line. And you're, again, not seeing that with that highly tax-producing cell line. And what's interesting, again, here is that ATLED only expresses the viral protein HBZ. So it's starting to hint at possibly a role HBZ would have in kind of pushing out these exosomal immune checkpoints. We also did a function to see whether or not these immune checkpoints that are on the surface of these exosomes can actually bind to its associated ligand. And since BTLA was the highest analyte that we saw within our assays, we used that as a representative ICP to then see whether it could actually bind to its biotinylated ligand. And we saw that with OSP2 and ATLED, we had a high level of binding compared to just Jurkat or MT4 cells. Another study that we did for characterization purposes was to see whether different cargo is present in larger versus smaller extracellular vesicles. And we saw, so I used a differential centrifugation method where you're basically using speed as a way to separate larger vesicles from smaller vesicles. And then we just confirmed the sizes with NTA analysis. And we saw that the majority of these immune checkpoints like BTLA and PDL2 was really only present in that 100K or that exosomal fraction, where you're not really seeing their presence in those larger 2K or 10K fractions. Another interesting find was that HBZ was also uniquely only present in that exosomal 100K fraction, which it was something that hasn't really been seen before. We also quantified some of the analyte levels in our fractionation data just to reconfirm whether they're just uniquely present in that 100K fraction or versus the larger vesicles. And for the most part, we saw that the majority of these analytes were present in that exosomal fraction. Lastly, just to do a final characterization of the cytokines that were associated with these isolated exosomes, we did a MSD analysis with a cytokine array, just looking at what overall phenotype these infected cell lines have, and particularly what kind of cytokines these exosomes are carrying. And interestingly, we found that both in just the supernate and the free soluble as an exosome depleted and in exosome associated cytokines, we saw high levels of interferon gamma. And interestingly, a study from 2017 from a cohort in Iran found that there's actually a positive correlation between the levels of HBZ expression as well as proviral load tied together with interferon lambda expression in HAM-TSB patients. Now, interferon lambda is a type 3 interferon, but it's unique in the sense that it does function very similar to interferon gamma, except that while interferon gamma is an acute response, interferon lambda is a more prolonged and sustained release. So what I decided to look at is whether there was high levels of interferon presence in our OSP2 cells, and we do see a substantial amount of interferon within our OSP2-like systems. So then I wanted to see whether interferon plays a role in overall changes in immune checkpoint levels, and we saw that although interferon played a more protective role with the Jurkat, ATL, and MT4-like cells, where you're seeing an increase in co-stimulatory molecules, it had an inhibitory effect in the OSP2 cell system, where we're seeing an increase of inhibitory immune checkpoint receptors like PDL2, LAG3, and TIM3. Next, I also looked to see whether using an antiretroviral drug would in any way affect the overall release or presence of these soluble immune checkpoints. And what we saw again was that with that HAM-TSB-like cell line, when we treated it with antiretroviral drugs, one targeting the retroviral protease, another the reverse transcriptase, we again saw this loss of inhibitory immune checkpoint molecules like CTLA, LAG3, and PDL2 only in that HAM-TSB-like cell system. And when we looked at the viral protein expression associated with antiviral treatment, what we ended up seeing was that HBZ actually was diminished across the board in all three cell lines that also had that loss of immune checkpoint expression. And we could see that it wasn't really an artifact of death because we didn't see any significant toxicity with the treatment. Interestingly, I decided to then just look at the overall surface expression of immune checkpoints on these T cells. And we found that, again, ATLED and OSP2 actually had a high level of several immune checkpoints being expressed on its surface. which really kind of, I guess, directed me towards looking into HBZ because ATLED really only does express HBZ as the viral protein. So I decided to use a lipoeffective system with a lentiviral vector that targets HBZ to knock it down, as well as a CMV-based promoter flag tag HBZ overexpression vector to see whether either knocking it down or overexpressing it would have an effect in overall immune checkpoint presence. And what we saw was that with the knockdown and overexpression, we saw a significant decrease and increase of PDL2, LAG3, and BTLA in our exosomes, which clearly linked the role HBZ has in pushing out or secreting out these immune checkpoint molecules. Now, just to kind of get an idea of what could possibly be the mechanism associated with HBZ secreting out these immune checkpoint molecules on these exosomes, I looked into two pathways. One is the endocrine growth signaling association pathway, which basically is known to play a significant role in just secreting out growth factors in exosomes that promote survival and plasticity, and is known to be elevated in HTLV1 infected cells. And we saw that TRKB in particular was both significantly decreased and increased with the knockdown or overexpression in both of our ATLED and HAM-TSB-like cell systems. I'm still navigating how that's exactly happening because from what we know about HBZ, it is a regulatory protein and not a transcription factor. So it has to be binding to another transcription factor and then kind of acting on it somehow. I also decided to look at the escort pathway just to see whether we're seeing some kind of engagement there with the HBZ protein as well. And only in that HAM-TSB-like system with the knockdown and overexpression did we see significant changes in the expression of HRS, ALEX, and VPS4. Again, this kind of alludes to perhaps HBZ kind of interacting with transcription factors that are associated with the expression of these escort associated genes. And right now that's where I am in my project, kind of trying to figure out which specific transcription factors HBZ may be interacting with in the context for both escort-related genes as well as that TRKB-BDNF autocrine signaling pathway and how that in turn is affecting the release of these immune checkpoint molecules. So that's one of the future directions, just specifically kind of teasing out and understanding the mechanism in which HBZ is really modulating these two pathways and understanding which transcription factors are involved, as well as seeing kind of the effect mechanism on healthy T cells and dendritic cells, whether or not these exosomes that are carrying these immune checkpoints are really affecting activated T cells. Is it contributing toward a more immunosuppressive response instead of engaging in active T cells response, which is what we would hope for in a retroviral infection. With that, I just want to acknowledge my lab, current and former members, as well as my thesis committee, all of my collaborators, the PenVet Core for helping me with all of my exosome isolations, as well as my funding sources and the T32 training grant that's helped me be able to do this project.
Really great work. The question's related to the source of the exosomes. So with this disease, it's if you were to isolate exosomes from neuronal cells versus ones you're working with from peripheral cells, yes, to the function, would you predict that you would find the same downstream effects on these?
I actually don't think you would. There's been a lot of studies that have shown that depending on the source of your exosomes, you're getting either a more suppressive or a beneficial effect. There's been studies that shown that exosomes derived from mesenchymal stem cells have a more regenerative effect when it comes to neuro regeneration or spinal cord injury. It's also seen to be more protective in Alzheimer's and Parkinson's patients, but we've seen that in tumor-associated exosomes or exosomes from infected cells in general have a more tumor immunosuppressive effect.
Okay, and the samples that you were using usually they're from peripheral blood from ham-TSC patients?
Yes.
Okay, got it. And then my follow-up question is, so because the exosomes are taking on these immune checkpoint inhibitors, I mean, I'm also just thinking some of them are heme molecules. So does that enhance cell to cell? Would having an exosome with some of these molecules on the surface enhance cell-to-cell contact and maybe transmission of the viruses in some way?
So there hasn't been studies that shown transmission, but there's definitely been studies that shown that when you're exposing just healthy cells to exosomes derived from an infected source, they do tend to start to kind of adhere to each other. So if your exosomes potentially carry an infectious agent, they can kind of promote viral spread, because we're seeing that just exposing like healthy PBMCs to like jerked isolated exosomes, they start to kind of stick together and form clumps, which would then help propagate infection if there was any present.
I'm curious about the interferon gamma using those. If they were all of them having those more immune checkpoint, maybe dampen the response. Why is there more interferon gamma in those cells? And when you knock it down in the HCC system, do you see changes in interferon gamma in those cells?
No. So for the first part of your question, we're not. So ham-TSB is known to be a more pro-inflammatory disease. So there are elevated levels of interferon present in patients. So we're just seeing that with our OSP2-like cells. With the knockdown studies, we actually saw that there wasn't a change in interferon gamma levels associated with HBZ, but that could be that the viral protein tax, which is also active in ham-TSB, could be contributing to the interferon gamma story more so than HBZ. It could just be completely independent.
I have a cool question. So, you mentioned that there was some ER protein that is a marker of non-exosomes, like it's retained in ER. Is there a protein that you can probe for in your Western blots that is a good, like, positive control? Yes, these are exosomes. I noticed you were using beta actin as your loading control, but I think that is like a cytoskeleton protein. So I don't know much about exosomes. I don't know what's standard, but I was just wondering, is there one protein that's in all exosomes or anything?
So there isn't one specific protein, but usually the class of tetraspanins from CD80, CD9, and CD63 are at least 2 out of the 3 are commonly found. So most characterization studies, you would hope to see at least 2 tetraspanins in your isolations, but those tetraspanins are also present in your just cell lysates as well. So the only marker that can really differentiate is whether you're seeing proteins that are only known to be associated with the ER in your cell lysates and not in your exosomal samples.
Okay, thank you. Any other questions?
I guess any HIV drug doesn't work against these HBV.
No, so there's been a lot of preclinical data that was promising, but in the clinical setting, so far there hasn't really been any HIV associated drugs that shown promise.
I think we're good. I believe we're taking a break until 5:30 for our keynote speaker. So people on Zoom, I guess, hold your breath until then. Thank you.
Thank you. Oh, turn it on.